It's a pleasure for me today to introduce Derek Lee, who is a doctoral student in my lab working on cancer metabolism. Derek just published a major paper in the field showing the strongest evidence to date that in addition to glucose that is known to be fermented to lactic acid in cancer cells, Derek has very strong evidence showing that glutamine, the amino acid glutamine, is also fermented to succinic acid through the glutaminolysis pathway in the mitochondria. People in cancer have known for a long time that glutamine is a major fuel for driving the dysregulated growth of cancer cells, but it was not known the mechanism, the process by which glutamine is driving this. Derek has unambiguous evidence that glutamine is actually fermented in the mitochondria through what we call mitochondrial substrate level phosphorylation. And he's doing an excellent job in defining this process more and more. And we're very proud of what he's been able to accomplish that will move the cancer field forward in the right direction.
So I'm Derek Lee. I'm a fifth-year PhD candidate in Dr. Seyfried's lab, and I recently wrote the paper on amino acid fermentation. I think to understand what a cancer cell is, at least in our perspective, we have to understand what it is in the field's perspective first. So I would say the field looks at the accumulation of somatic mutations over time, maybe a bad luck hypothesis, and they would say that once you acquire a certain number of somatic mutations, the cell will then start to proliferate and go back to its default setting of proliferation. However, we look at it from a different perspective. We say it's very focused on the mitochondria rather than the nucleus and acquired somatic mutations. So we say that, and I should also add that once you acquire somatic mutations, those somatic mutations then alter the metabolism. So that's why we're seeing the lactate and the succinate that we're seeing. So we have a different view. We're saying that there's first damage to the mitochondria, and importantly that it's chronic and sublethal. If it's too acute of damage to mitochondria, then you'll just kill the cell. The cell will abort, it'll go through apoptosis, which is kind of a slow programmed cell death mechanism, and the cell will just die, and you cannot get cancer from a dead cell. So we look at the metabolic perspective. So we see there is damage to the mitochondria. That causes a fundamental change in energy metabolism because the cell is now getting less ATP out of the mitochondria, and that sends signals such as ROS or retrograde signaling to the nucleus. ROS are both carcinogenic and mutagenic, which means that those will then create mutations in the nucleus, and then you see all the downstream effects and the hallmarks of cancer that you regularly see. So it's a bit of a chicken and an egg situation. I think a lot of the field says at first it is the mutations that cause the metabolism. We're saying it's the metabolism that's causing the mutations and then all the downstream effects.
And Dr. Seyfried would often say, everything starts with energy. You don't have energy, you don't have a human being, you have no life. So let's go to the source of energy production to figure out where to begin the conversation. And therefore, he arrives at the mitochondria because it's intaking oxygen and generating ATP.
Yeah, I mean, at the end of the day, energy is everything. Every living thing in the biosphere requires energy to be alive. So there's a ton of pathways you can target in a cancer cell to try and kill it. However, we think if you go for the fundamental difference in a cancer cell versus a normal cell, based on the theory that Dr. Seyfried has described and I've described previously, we think that one of the fundamental differences in all cancer cells, regardless of a BRCA mutation or a P53 mutation or any kind of mutation you want to say, we're saying there is a fundamental difference in metabolism. So if you can target that fundamental difference in metabolism, you can more effectively treat a larger range of cancers than a more specific approach by targeting mutations.
And if you were to put it simply, what is the difference in the metabolism?
The difference in metabolism between a cancer cell and a normal cell is how it produces energy. That will cause all the downstream effects that show other differences in metabolism. For example, you might see greater cholesterol biosynthesis in a cancer cell. Now, is that the first change in metabolism you see during carcinogenesis, or is it perhaps downstream of energy production? Like we've said in our theory, we think that most of these effects are downstream of energy production, so we want to target that, and that's what we refer to when we say fermentation. Fermentation is ATP synthesis without oxygen. So in a very broad term, a normal, quiescent, healthy cell in the body will take in oxygen and glucose. Glucose will break down into pyruvate in the cytoplasm. Pyruvate will then be transferred into the mitochondria. It will go through the TCA cycle and then go through the reducing equivalents. Will go through the electron transport chain, produce that gradient in the inner mitochondrial membrane, and then you will then translocate those protons that are in the inner mitochondrial membrane and produce ATP in a very highly efficient, highly controlled manner. What we're saying is in a cancer cell, that efficiency in that mitochondria is lost. So instead of taking in oxygen and taking in glucose and going through that whole pathway I just described, you'll take in very large amounts of glucose, convert it to pyruvate, and then convert it to lactate. And that lactate will then be expelled outside the cell, which is why you see this huge acidification in the microenvironment, which is a common hallmark across all cancer cells. Now, an important feature of that when we're talking about energy production is that if you look at it on paper, that glucose to lactate pathway I just described is only 2 ATP, whereas oxidative phosphorylation is 32 to 36 ATP depending on how you do the calculation. So naturally you would ask yourself, how can a cell even live if it's only getting 2 ATP per glucose molecule or 36 when the highly efficient respiration? The way it does that is cancer cells take in a huge amount of glucose. There are various estimates in the field, but you could say anywhere from 20 to 40 times as much. And if you do the multiplication, it then can compensate for the decrease in energy efficiency in the mitochondria. And this is a hallmark of all cancers. This is actually how we detect it in the clinic by using FDG PET scans. Cancer cells take in so much of this fluorescent glucose, you can see it on a PET scan, and that's what we use for advanced cancer detection. So it's very common amongst all cancers.
I picked out this quote from the paper. It says, "Indeed, accumulating evidence shows that the dysregulated growth of cancer cells is driven in large part by altered mitochondria energy production." So can you explain what is the scientific evidence that you discovered, that the paper revealed, that the experimentation revealed, that led to that conclusion?
Yeah, so a lot of this goes sort of into the background of why we think there's an issue with energy metabolism. So Dr. Seyfried in the past has looked at the structure, function, and number of mitochondria. And it's not that there's always just a few mitochondria in there or that they're all broken all the time, but it seems like there's a clear trend of a difference in those three factors of mitochondria between a cancer cell and a normal cell. If you look back at some of his previous papers, he has entire tables that go through all the major cancers, and it seems to be a common trend amongst all of them. So that's why we tend to look at things like the Warburg effect, which is a very well-known aspect of cancer metabolism. We focus on the fact that it may be due to deficits in energy metabolism. So when the mitochondria can't effectively produce energy through respiration or oxidative phosphorylation, due to damage in the mitochondria, it then falls back on things like the Warburg effect, and that's why you see acidification in the microenvironment due to both lactate, and what my paper now tries to explain is that it could also be due to a little bit of succinate as well.
So it says in the paper, the aim of the research was to determine, one, if amino acid fermentation could contribute to ATP content in mouse and human glioma cells, and two, if oxygen consumption was an accurate marker for ATP production through OXPHOS in these cells. So what did you learn with respect to these two aims?
So the reason we really beat the drum on amino acid fermentation is we're really trying to look at the mitochondria and see where the energy inside them is coming out of. Traditionally, it's thought that all of the energy coming out of the mitochondria is through oxidative phosphorylation, but there is a substrate level phosphorylation, which is what we refer to when we're talking about fermentation, that's also in the mitochondria. And naturally, with having two sources of ATP within the mitochondria, it can be difficult to distinguish the two of them. And largely, mitochondrial substrate level phosphorylation has been, put to the wayside a lot in research. So my paper is trying to focus on that aspect, kind of disentangle them a little bit. So what I found in my paper is that, yes, although on a per mole basis, you might only get, very little ATP coming out of mitochondrial substrate level phosphorylation, there's so much of the upstream metabolite, in this case glutamine, coming into that pathway that it might actually contribute a lot to that total fraction of mitochondrial ATP. And that's why we try to focus on it, right? Because in a therapeutic context, if we're looking at targeting mitochondria in cancer cells, a lot of investigators have used drugs to inhibit the electron transport chain or the mitochondria in general. And if the logic there is to inhibit mitochondrial ATP, you might be skewing towards targeting OXPHOS. You can't really do that in the therapeutic context, because once you get out of the cell culture plate and go into humans, you're now going to start damaging the normal cells. So if my paper can at least show partially that some of that ATP is coming from mitochondrial substrate level phosphorylation as opposed to oxidative phosphorylation, which is pretty much solely what normal cells rely on, it might give less incentive to try and target OXPHOS in cancer cells, which we actually know has had pretty devastating outcomes in the clinic. So oxygen consumption is what investigators have relied on mostly to use as a proxy for mitochondrial ATP content. So what we'll do is we'll measure the oxygen consumption coming into a cell, put a drug that will inhibit the electron transport chain, see the drop in oxygen consumption, and look at that difference and try to equate it for how much energy is coming from respiration in a cancer cell. Now, in a normal cell, I think you can do that because normal cells are pretty much getting all their energy from respiration, but we think that might be misleading in cancer cells. There's several reasons for this, one being that not all oxygen in a cell is being used for oxidative phosphorylation. Oxygen is the terminal electron acceptor of the electron transport chain. However, there are also cytoplasmic reactions that use oxygen. There's also uncoupling in the mitochondria that can cause the oxygen input to not equate to the ATP output that investigators assume. For example, if you break down a substrate in the mitochondria, you might cause a buildup of protons in the inner membrane, which is kind of the driving force behind oxidative phosphorylation, but that doesn't always come through complex 5 or ATP synthase. So if it comes out of the inner membrane through a different mechanism, such as an uncoupling protein or even back leak through the other complexes, you've now expended oxygen as the terminal electron acceptor, but you haven't gotten ATP out of it. So because of those two reasons, as well as a few others, you can't always use oxygen consumption as a pure surrogate for mitochondrial ATP production.
So oxygen is going into the cancer cell, but it's not necessarily being used for the production of ATP.
Yes, and some of it is, right? We're not saying that there's no OXPHOS in a cancer cell. However, we are saying that it does seem to be damaged, or the term we like to use a lot is insufficient. That's why we see this compensatory glycolysis, or in the case of my paper, we're now also investigating glutaminolysis as well. When you have damage in the mitochondria, like I said, it will send out these retrograde signals to the nucleus. There's a lot of cross-communication between the mitochondria and the nucleus. When the mitochondria gives this retrograde signaling, it will signal to the nucleus that you need to upregulate fermentation mechanisms. So some of those might be c-Myc, which is a common oncogene that people describe in the literature. That will bring in more glutamine to be fermented, whereas it will also upregulate HIF-1 alpha, which is probably one of the most well-known master transcription regulators of glycolysis. So once the mitochondria tells the nucleus that there's insufficiency here, there's damage here, the nucleus will upregulate these fermentation pathways, and that's what will help facilitate this high throughput, inefficient form of glucose breakdown. The difference between the CT2A, which is a mouse cell, and the U87MG, which is a human cell line, is the basal metabolic rate. So the basal metabolic rate describes how quick the general metabolism in a given cell is. I think it was first thought that it was by organism to organism, but it actually seems to be intrinsic to something within the cell, because even in cell culture, you see these big time differences between a mouse cell like CT2A or a human cell like U87 and how long it takes them to run out of fuel, for example. So that's kind of what we wanted to describe, at least show with that comparison, is that between a human and a mouse, even when you take everything else away from the organism and just focus on the cell, there is this difference in basal metabolic rate. So this is really important when you start looking at mouse experiments. You know, a drug that worked over the course of 6 hours in a mouse might take several days in a human. So it just kind of shows that the scaling factor between a mouse and a human are different. For a limited time, a cancer cell can survive on just one of them, but eventually they do need both of them, and that's for a host of reasons. We primarily focus on the ATP reason, but there are also other pathways that glucose and glutamine feed into, you know, things like glutathione production or 1-carbon metabolism, which are a bit in the weeds. But the point is that by itself, they cannot survive off one of those metabolites alone. Now, when we talk about a therapeutic context, you will never get one of those metabolites to zero by itself. So again, this is kind of a benefit of using cell culture to investigate this, is that we can look at that sort of experiment. But if you cannot reduce one or both of those metabolites to zero in vivo or inside a person or a mouse, that means you have to target both of them to lower them both simultaneously to try and get this killing effect.
Though it could be effective to lower, let's say, glucose because it's a little easier to target dietarily.
Yeah, it's definitely great to lower one of them. It's optimal to lower both of them.
This data show that glutamine alone can maintain limited ATP content under severe oxygen deprivation. Viewed collectively, these findings present strong evidence showing that glutamine can support ATP production in mouse and human glioma cells through mitochondrial substrate level phosphorylation in the glutaminolysis pathway.
Yeah, so one of the difficulties I described of investigating mitochondrial substrate level phosphorylation versus oxidative phosphorylation is that glutamine actually feeds both of them in normoxia, which just means a normal amount of oxygen in the air, what you and I breathe right now. What I did was I put my cells in hypoxia. So by putting them in hypoxia, which means 0.1% oxygen, it's now inhibiting the availability of oxygen. So now it's another argument to take oxidative phosphorylation outside of the equation. So when I put my cells in glutamine alone inside of hypoxia, I was still able to find bioluminescence, which means that there is some effect from mitochondrial substrate level phosphorylation, which you wouldn't always be able to determine in normoxia because, like I said, glutamine can feed into both pathways. So it's a way to kind of separate the two without using a drug at least.
And could you explain to us what mitochondrial substrate level phosphorylation is? I know we often think about it in contrast or in opposition to oxidative phosphorylation, but what's happening in the cell that we call MSLP?
Yeah, so MSLP is a specific reaction inside of the glutaminolysis pathway. So once glutamine comes into the cell, it will be converted to glutamate. It'll take off one amide nitrogen. It'll go from glutamate to alpha-ketoglutarate, take off the second nitrogen group, and then now you have alpha-ketoglutarate. Alpha-ketoglutarate is part of the TCA cycle, which we're very accustomed to in biochemistry diagrams and normal cells. Once you have alpha-ketoglutarate, you're converted to succinyl-CoA. From succinyl-CoA to succinate, which is the next reaction in the TCA cycle, you actually produce an ATP molecule in that reaction. Sometimes you also produce GTP depending on the isozyme or the variant of the enzyme that facilitates that reaction, but GTP can rapidly be converted into ATP. So when we refer to mitochondrial substrate level phosphorylation, we're specifically referring to the conversion of succinyl-CoA to succinate and the ATP that's produced there. Now, what happens to succinate? In a normal respiring cell, succinate will continue along in the TCA cycle and go from succinate to fumarate to malate and oxaloacetate and continue in the TCA cycle and eventually be diverted towards a different pathway. What we see in fermenting cells or cancer cells, as is in the case of my paper, we see that succinate not only breaks from the cycle and you find it in the cytoplasm, I was actually able to measure it outside of the cell entirely into the extracellular matrix. So you would never expect to see succinate in the cytoplasm, let alone outside the cell, if you were using primarily oxidative phosphorylation as an energy source.
I'm thinking about the hundreds of people that are listening to us, watching us on YouTube. trying to interpret the papers that are being written and published out of the lab here. And just what do you think the whole movement needs in order to bridge the gap between the production of literature here and the people at home?
It's very challenging sometimes to take a specific experiment from a specific paper and try to make an actionable approach on that. That's why we do both the data papers, like the one I just published, and we also try to write review articles to try and take some of the field together and do more actionable approaches. So I think if you're looking at the cancer literature and you want to find an approach for yourself or someone else, I think it's important to find out what variables can you manipulate. For example, when we talk about glucose and glutamine, you can inhibit the amount of glutamine or glucose coming into your body by not eating carbohydrates or sugar, such as a ketogenic diet. Whereas glutamine, at least in the diet, you can't avoid glutamine. Glutamine is a fundamental amino acid that is in all types of food. So that's where, unfortunately, we need to use things like drugs or different compounds to inhibit the utilization of glutamine. So it's not that you have to understand every single aspect of a scientific paper, because it is very challenging, but I think it's important to think about what are the takeaways from that paper and what's actionable for you. I think it's really important to evaluate what stage your cancer is at. For example, stage 0 or stage 1 cancers, there's a very high survival rate for those cancers. You don't always have to go on a 14-day water fast to deal with a stage 0 cancer, but you might want to do a ketogenic diet if you don't want to do a low dose of chemotherapy or a surgery to extract it. It's kind of about balancing the risk to reward ratio for a given treatment. There's no treatment on the planet that has no risk, but you have to balance what your stage is versus what treatment options you want to approach. I think something like press pulse protocol is something that's more for stage 4 cancers, because you've likely exhausted all other options and maybe the first line chemotherapy didn't work, or it only stopped the tumor for a little bit and now it's come back. So press pulse protocol can now introduce a new modality to try and fight that cancer, whereas maybe a second line or third line chemotherapy may not have helped. But again, if you have a stage 0 or a stage 1, maybe doing something like a hybrid of metabolic therapy plus some standard of care can be applicable, and you can see great results without having to do a full-blown protocol or something more drastic like that. I think unfortunately I found myself in that situation. I would definitely implement the press pulse protocol, and I think I do have to say that to some level, but I do truly believe in it, right? Because I've spent a lot of time investigating what a cancer cell actually requires, and I know a lot about the differences between the requirements of a cancer cell versus a normal cell, which is kind of what my paper went into, right? Although I didn't have a normal cell to compare it to, we do those studies in the lab with other people in our lab, and I'm pretty confident that if I can restrict glucose and glutamine in myself, I would have a much better chance against cancer than someone who did not. Now, do I strictly have to do press pulse protocol by itself? No. I think you could do a press pulse protocol or metabolic therapy or ketogenic diet in conjunction with something like a low dose chemotherapy. Not all chemotherapies are blisteringly toxic to the body, but at high doses they can be. So one of the benefits of doing a ketogenic diet is that sometimes you can reduce the amount of chemotherapy you need, or maybe in some cases avoid it entirely. But I think the first adoption for ketogenic metabolic therapy or press pulse is a hybrid approach, and I think that's most sustainable for most people, because you will have the support of your oncologist, and you won't have to deal with bit of an argument back and forth between an oncologist or the primary literature you're reading, which is honestly a very important consideration when you're in this situation. You might be very bought into what we write about, but you don't want to have an argument with your oncologist about it. So I think what a lot of people do is a hybrid approach, and we've seen very successful results as a result of that. So you don't always have to do one or the other. Now, I personally am a bit stubborn, so I think I would err on the side of doing ketogenic metabolic therapy or press pulse by itself first, but I would never rule out taking a small dose of chemotherapy to augment the state of nutritional ketosis that I'm now in because I did metabolic therapy as a primer for that low dose chemotherapy. Yeah, so cancer prevention is really important because if you can just avoid dealing with the problem in the first place, you never have to go to those more severe options. What I do is I try to eat a low carbohydrate or ketogenic diet in general, and I also work out a lot because I think if you put these minor stresses on your body, such as exercise or not a huge influx of carbohydrates, I think these can reduce the chance of getting risk factors that will lead to cancer down the road. So for example, with food, if you're eating a highly processed carbohydrate diet, that might increase your risk of diabetes, and it in fact will. Now, diabetes and obesity is now essentially the number one risk factor for cancer. So if you can avoid getting diabetes or becoming overweight, such as by avoiding a highly processed carbohydrate diet, then you'll have a lower chance of getting cancer, and that will help you prevent it. Now, I also always stress to people the importance of exercise. If we want to look at glucose and glutamine, for example, like in the case of our work, muscles function as a sink for glucose throughout the day, and especially when you're actively doing exercise. Your muscles burn glucose very fast, and that can help reduce your glucose levels in your blood so you don't have excess glucose in your blood all the time. And another really important aspect about glutamine is that we now know that exercise can reduce glutamine in your body. There was a study, or several studies actually, that very high intensity exercise, so not just walking around or going for a light jog, but maybe sprinting or very severe weightlifting, can actually reduce glutamine in your body, the concentration in your blood, for several hours. Now, that might not be enough to completely starve the cancer cell entirely, but if you're doing something like that in conjunction with a ketogenic diet, you're now effectively reducing both glucose and glutamine, pulsing it or pressing it, depending on which modality we're talking about, and that can help both prevent cancer induction or potentially help treat it as well.